Welcome, everyone, to a special celebration for a very special occasion. We are here to celebrate the latest member of the College of Chemistry faculty to receive a Nobel Prize, Jennifer Doudna. And as I've said before, Jennifer's work and spirit embody what is best about the rich scientific tradition and collegial culture of the College of Chemistry in UC Berkeley. And as you all know by now, the discovery of CRISPR-Cas9 by Jennifer and Emmanuelle Charpentier, the first two women to ever share the Nobel Prize in chemistry, has already revolutionized biological research by allowing scientists to rewrite the code of life in any organism with unprecedented efficiency and precision. Furthermore, the groundbreaking power and versatility of CRISPR-Cas9 has opened up new and wide-ranging possibilities across biology, agriculture, and medicine, including the treatment of intractable diseases. But as you all know, Jennifer is much more than a brilliant scientist and educator. She is an incredible colleague who is extremely supportive of our college and a wonderful representative of our college, an inspirational role model, an entrepreneur, and a leading advocate for the ethical use and regulation of CRISPR technology. Jennifer, we are so lucky to have you as a member of our community. And today we're especially lucky to have this opportunity to engage with you in a unique Q&A with questions from our students. So without any further ado, I'll now turn things over to Matt Francis, the chair of the chemistry department.
Hi, everybody. Thanks for joining us this afternoon. So it's my pleasure to kick off the event. Let me echo the words that Doug said about how proud we are of Jennifer Doudna and all of her contributions, and also just what a terrific colleague that she's been to all of us. A little bit of a personal side of this on what happened when she won the Nobel Prize. So Doug and I, by tradition, we are awake at 2:45 a.m. We were both logged on to the live broadcast when they announced the Nobel Prize. I cannot speak Swedish, but as soon as they started saying the names, I could figure out that this was going to be the prize. This is going to be Jennifer's year. And so we were very excited. It turns out we, as I understand it, also found out about this before Jennifer did, because I understand that she slept right through it and actually got a call later in the evening from Nature, or later at night by Nature magazine to ask what her opinion was. And she's like, well, who won? Anyway. So we followed that up with a press conference the next morning, and then in the afternoon we had another meeting that had the faculty of MCB, the faculty of chemistry, and her research group on it, and many other members of the IGI. And I guess from a personal perspective, I'll echo something Doug said, that to me that event just really represents what's so special, what really makes Berkeley a different place than every place else. It's just everyone was so happy. Everyone was just delighted with her success. Everybody had all kinds of stories to share about what a terrific colleague she was, a collaborator that people have had, collaborations they've had with her. And it was just a really, really neat event. And what we really wanted to do was to have another event that really involved the entire College of Chemistry in it, and also one that really focused on the students and the staff. As faculty members, we often get to ask Jennifer questions here and there in meetings, but we really want to provide an opportunity where our community, broadly speaking, got to ask questions, anything they wanted to know about CRISPR-Cas9 or anything they wanted to know about Jennifer's path to success. And so what we decided would be a great format for this is we have two student moderators, two graduate student moderators who are going to MC this event and ask the questions today. The first is Lydia Boyk. Lydia is a graduate student in Dan Nomura's lab, and she works on chemoproteomics approaches to developing or identifying new targets that can be used for cancer therapies. And then we also have Max Sosa, who's a graduate student in Michelle Chang's lab, and he studies secondary metabolism in bacteria with the goal of identifying new pathways that could be used for antibiotics. And so thank you both of the students for joining us today. Thank you, Jennifer, for sharing this event with us and giving us some of your time. With that, I will turn it over to Lydia and to Max to ask the first question.
Hi, everyone, and hi, Jennifer. I'm really happy to be here today to help facilitate some questions from students. Like Matt said, I am a third-year graduate student in chemistry. So Jennifer, the first question that we have is, could you tell us a little bit about CRISPR-Cas9 and the history of this discovery so that people in the audience who might not know as much about your work can have a brief introduction?
Sure. Well, first of all, I want to say hi to everybody that's on the call today, and a huge thank you to the College of Chemistry, to Dean Clark, Chair Francis, and to you, Lydia and Max, and Rachel's here as well, and Sarah, for setting this all up and hosting it. It's really an exciting opportunity for me. And I've been a member of the College of Chemistry for 18 years, going on almost 19 years. So I moved to Berkeley in 2002. And one of the big attractions for me, and I was moving from Yale, where I had started my faculty career, a big attraction was that at Berkeley, chemistry, the College of Chemistry, considers chemistry to be a very broad field. So it includes people doing work like I do, which is studying biomolecules and figuring out how they work. And that was really the basis for getting involved in the CRISPR project, because when I got to Berkeley, I was already very interested in investigating RNA molecules that control gene expression in cells. And we'd done a lot of work on very tiny RNAs called interfering RNAs that have that behavior, that activity in human cells. And I was intrigued with the discovery of a pathway called CRISPR that was hypothesized to be an adaptive immune system in bacteria. This was work that Jill Banfield here at Berkeley was doing at the time. So we started to investigate the molecules involved in this proposed immune system in bacteria, thinking that there was evidence that it was an RNA-guided system. And I thought it would be fascinating to figure out how bacteria might have the ability to adapt to viruses using an RNA mechanism, RNA-mediated mechanism, and how that would compare to pathways like RNA interference in eukaryotic cells. And so that was really there was not really a bigger vision than that. It was really just, how does this work? And this sounds really like a very interesting twist of evolution, that bacteria would have evolved this kind of a pathway. And then, so the project started very, very, very small in my lab. It was just a couple of people doing experiments, and a couple of chemistry students. So Sam Sternberg was a chemistry student who came to the lab early on and started working on the mechanisms of Cas9, or the CRISPR-Cas proteins. And then we started a collaboration with Emmanuel Charpentier's lab in 2011 to study a particular protein, Cas9, that was implicated in this bacterial immune system, to figure out how it worked. And that led to experiments that uncovered its activity as a double-stranded DNA cleaver that was RNA guided, and not only guided by one RNA, but two RNAs. It was dual RNA guided protein. And once we understood that mechanism, it was possible to simplify it as a single guided RNA programmable protein that could be harnessed as a cleaver in any type of cell. So you could use it to cut DNA in bacterial cells, but also in the genomes of eukaryotic cells. And that then immediately implicated it as a technology for genome editing. So, I think CRISPR is a great story of the importance of fundamental science, of curiosity-driven research that many of us value here at Berkeley. I think, it's sometimes one can, when we're students, one can lose sight of like, why am I doing this? Why does it matter? And I do think that science is, one thing that's so exciting about science is that we don't actually know where it's going and what the outcomes of our experiments are going to be. And we just need to, pursue our passions and keep an open mind to what our results might mean.
Awesome. Thank you for that beautiful answer, Jennifer. And great to talk to you again. As Matt earlier introduced me. I'm Max Sosa, also a graduate student in the chemistry program. And kind of following on that last note, I guess, you don't know where your experiments are going to lead you. These kind of experiments have led to some big ethical questions. So our next question that we have received is, In the 10 years since you've started this work, has there been a shift in how we think about ethics in science, and how did that impact your work?
Well, I think, I don't know if there's been a shift in how we think about ethics in science, but I do think, well, there's certainly been a shift for me in the sense that, me personally, in the sense that I think like many of us, one doesn't necessarily imagine that experiments you're doing in the lab could have broad societal, ethical implications in the long term. But I do think that, I've certainly come to appreciate that we always have to be open to that or thinking about it in our work, because every now and then that's exactly what comes along. And so this is the case for CRISPR. And just for folks that are on the call today, just to mention a couple of things. So, CRISPR is a technology that allows precision changes to be made to the DNA in cells of organisms. And that includes embryos, it includes germ cells, and that kind of genetic change is heritable. So those changes can be passed on to future generations. And so that potential use of CRISPR, especially in humans, has really triggered a kind of a long-standing global discussion now about the technology that I've been involved with. And I have to, again, just really give a big shout out to Berkeley and credit to our great university for being a place that fosters that kind of thinking. And we have many people in our community here who think about bioethics, they think about the law, they think about the psychological implications, the historical implications of technologies. And I've benefited from all of that, really. And so it's been just extraordinary to be here working in this community where you can do absolutely cutting edge research, but you also have access to people that think at the cutting edge of all these other fields that sometimes are intersecting, and that's certainly the case with genome editing.
Thank you. I guess looking more towards the application side of CRISPR, how soon do you think we could see a future where nucleic acid targeted small molecule therapeutics become the standard for directed medical care?
Well, what's amazing about CRISPR is that it's not even a 10-year-old technology yet, and yet it's already in clinical trials for certain diseases. So one example is sickle cell anemia. So this is a very well-known genetic disorder that involves a single gene, the beta globin gene, that contains a mutation in folks that are affected with this disease. And with CRISPR, we now have a technology that can, in principle, correct that disease-causing mutation or activate another gene called fetal hemoglobin that will suppress the effects of the mutated gene in beta globin. And so this is already happening. So we've had, over the last year, some announcements from both companies and academic groups that are working on this. And a patient named Victoria Gray has already received a CRISPR treatment for her sickle cell disease that has effectively cured her of this disease. And this is just, it's kind of mind-blowing how fast the technology is actually getting into the clinic and having real tangible benefit in patients. Now, that's great, that's very exciting, and I think it is sort of opens the door to many other uses for treating genetic disease. But it also raises the specter of cost and access, something that I think about a lot these days, because right now these technologies are, $1 million or more cost. So this is just not a cost that is, going to be sustainable in the future and will not enable the people that need this kind of therapy to get access to it. So that's something that at the Innovative Genomics Institute now here at Berkeley that we work on very intensively, is thinking about ways to develop the technology so that it will be affordable and accessible to those that need it. And I also just want to briefly mention that that the thing about CRISPR that's so, one of the aspects of it that's so interesting is that it is a very cross-cutting technology. So it's not applicable just to treating disease, but it's also very important in agriculture. It's also very important in synthetic biology, ways that we might be able to manipulate the genomes of microorganisms to make them useful in industrial processes and things like that. So, there are all of these other developments that are going on in the world of CRISPR that I think will also have really big impacts over the coming, 5 to 10 years, especially I think in agriculture.
Great, thank you. So I think it looks like we have some more questions about your path to becoming the Jennifer Doudna we know today. So could you please tell us about some of the significant challenges in your personal and research life that you had to overcome in order to reach the Nobel?
Well, let's start with, I grew up in a small town in Hawaii, and my parents were intellectuals. They were both, my dad was a college professor at University of Hawaii in literature. My mom was a history lecturer at the local community college. But nobody in my family, even in my extended family, I can't think of anybody in my entire family on either side, in fact, that was a scientist. So I didn't have anybody close by to talk to about science, and I certainly didn't know any working female scientists. I knew a few men who were colleagues of my dad at the University of Hawaii in science, and they were great. They were really interesting, helpful to me. But I didn't have any female mentors. So it was growing up, I sort of had this idea that it would be really cool to do science, and it would be kind of really unusual to do it as a girl, and so that was kind of in the back of my mind. And I had, like many of us, you have naysayers along the way, people who say, oh, that'll never work, or you'll never succeed, or this will, right? And you also have the voices in your own head, at least for me, that sometimes say those things. And so I had to find ways to just deal with that. And I think it's, for me, it's a combination of just a burning desire to figure out how things work. I don't know, I love doing that kind of work. So it's very compelling for me personally. It's just something I find to be fun. And also the joy of working with other people to make those kinds of discoveries. I found out very early on that, working in a lab is, we're not sort of working in a vacuum, right? We're actually working in a group of people, and you're talking about ideas, you're bouncing things around. Lots of things don't work, and you're trying to figure out why not. And, am I doing the best experiment I could be doing, or should I be doing something a different way? So, that was really fun for me also to kind of learn that that's kind of how science works. And I don't think you get that in any textbook. Like, I never did, really, right? So it's really just figuring it out by doing it and realizing that, wow, this is really cool. This is really fun. And so that kind of sustained me through a lot of the challenges that one faces in science. And then I would also just mention that I've had just great mentors along the way that helped me through difficult times, and sometimes unexpected. Like I've told this story before, but I had a French teacher in college who actually encouraged me to stick with my chemistry major when I asked her about the possibility of switching to French, and said, no, I think you should stick with chemistry. There's a lot more you can do with that degree. So, that was kind of important guidance at a key moment for me. And I do encourage students to, look for mentors like that, people who will support you and sustain you through the times that are challenging. When everything's going well, that's one thing, but how do you get through the times when you're struggling and experiments aren't working, and you're wondering, why am I doing this? And that's when you really need folks who will back you up.
Yeah, I believe that a lot of us can probably relate to how important it is to have really good and encouraging mentors and a community that we can work in. That's really encouraging as well. Thank you for sharing all of that. what is one piece of advice that you would give your younger self venturing into adulthood?
Well, I guess I would, what would I think I would probably tell myself to relax a little more. I don't know. It's easy, you know, hindsight is 20/20. It's easy to say that in retrospect, but I think that and again, I suspect I'm not alone in this, you know, that I've, when I look back on my life so far in my career, you know, I think I've often, you know, I get very intensely focused on something and struggling with a, you know, with a problem, and I'll just, you know, it'll be kind of all consuming for me. And sometimes what actually helps helps sort of with a breakthrough is taking a step back from it, you know, going out for a hike, or for me, you know, I love gardening, you know, just getting out in my garden and pulling weeds. I mean, you know, just, you know, stuff like that, that can actually, you know, open your mind to new ideas. And I've often found that to be the case for me when I'm, you know, struggling intensely with a problem and I can't figure it out and it's frustrating. And then you know, I go off and do something else, and I'll, you know, oftentimes I'll suddenly think, oh, wait a minute, you know, maybe I wasn't thinking about that quite right, or I could try something this other way. So I think that's something that I try to remember. I don't always listen to my own advice, though.
Definitely relate to that. It's hard to it's hard to listen to your own advice. You think you know what you're doing, and then all of a sudden you wish you would have listened to yourself, or at least someone else. So maybe we can backpedal, I guess, a little bit. You mentioned your mentors a little bit earlier, a French teacher you had. And so we have one question where somebody is recounting a beautiful graduation speech that you gave at the College of Chemistry graduation ceremony, where you mentioned that you had experience in your freshman general chemistry course. And when you took your first midterm, you were given mentoring advice after that. I'm not sure if this is the same mentoring advice, but if you could maybe recount that story for those of us who have not heard it, and maybe answer a more broad question, and who have been your mentors over the years, and how has that influenced your mentoring style?
Yeah, well, okay, I've got to try to remember the story now, but but I think it was generally something about the fact that, you know, so I went to public high school in Hilo, Hawaii, and you know, like a lot of people, I went from that environment where I, you know, didn't have to study too hard to do reasonably well, to Pomona College as a freshman, where, you know, I was taking general chemistry. And I was in with a lot of students that had, you know, gotten 5s on the AP chemistry test. Well, I'd never taken the AP chemistry test. My school didn't offer that, right? So we didn't have AP chemistry. So I was, you know, struggling to learn how to balance equations and, you know, all the fun stuff we do in general chemistry. It was tough. You know, it was tough for me. And you know, and our professor, R. Nelson Smith at Pomona, ran the class in a very distinctive way. So he had, you know, his lecturing style, and he loved practical jokes. So he would often play practical jokes on the students. And, you know, after every exam, at the end of the exam, you know, it's a 50-minute exam, he had this huge, you know, Beilstein, you know, book, and he would slam it down on the front desk and just break everybody's concentration. It kind of reverberated through the whole lecture hall, and that was how the exam would end. And then he would announce the high grade on the exam, you know, and then like a couple classes later, when the exams had been graded, he would announce the name of the person that got the high grade on the exam. So it was, you know, it was run in this very, you know, competitive way. And, you know, it was just kind of, oh, it's awful. For me, it was just kind of a horrifying experience. And struggling to you know, to get B's or B minuses on the exams. And, oh, it's just, you know. And so anyway, you know, getting through that class was a big challenge for me. And that's when I had the experience with my French teacher, where I loved French, and I'd been studying it through high school, and I was, you know, enjoying taking it in college. And I went to her and I said, you know, I don't know, I wanted to be a chemist, but I just don't know if I have it in me. I just don't know if I'm cut out to do this. And anyway, she gave me the pep talk I needed at the moment I needed it. And, you know, and then things got better, right? Because then I got into organic chemistry, loved organic chemistry. It was fantastic. It was just fun, right? You know, figuring out how to make molecules. I mean, it was like solving a little puzzle, you know, every day in class. And I don't know, it was night and day for me. It was suddenly I realized, oh, this is really fun and I can do this and I enjoy it. So it's you know, again, it's one of those stories that I try to remind myself of when I'm struggling through things now. Is like, okay, you know, things change, right? You struggle through something and then you know, you get through it and you get on to the next thing.
Thank you. Just now and a little bit earlier, you spoke about how a lot of your motivation comes from your desire to really understand thoroughly the scientific problems you're working on. And I was wondering, what other inspirations or motivations do you have to sort of wake up every morning and do research every day at the level that you do?
Yeah, well, you know, it really does come down to just wanting to understand the natural world in some chemical detail, and the, you know, it's kind of like Richard Feynman said, you know, it's the joy of finding things out. It's just that sense of discovery, of figuring out something that even as small or minuscule as it might seem, you feel like, well, I'm, you know, one of the first people, or maybe I'm the first person to ever know this little fact, and it seems kind of fun, you know. So I think that's part of it. Also, really enjoying the people that I'm working with, and, you know, fantastic group in my lab, people who are like-minded in their, you know, desire to understand molecular mechanisms and enjoy thinking about these problems and how to tackle them experimentally is also really fun. I think it's, yeah, I guess I just keep coming back to the, you know, the puzzle example. You know, like when I was growing up, my dad always liked to do crypto quotes. You know, these little, you know, encrypted quotations that you had to kind of solve the you know, and they, you know, they'd come out in the paper every day, and he'd do this little crypto quote. And we always had, I don't know, we always had all kinds of little puzzles lying around the house. We usually had a jigsaw puzzle going, you know, for somewhere in the house. And so I really grew up kind of with this, you know, sense that, you know, puzzle solving is a fun way to spend time. And I think that's a lot of what we do in the lab is, you know, we're really trying to solve a puzzle. We have a question we're trying to figure out, and we don't know how to figure out the answer to it. That's the challenge, is how do we design experiments that were going to give us an answer to this? And then, as you know, a lot of times those experiments either don't work, or we get data that don't make any sense. So we're trying to, you know, and that's the struggle part, right? You're trying to fit the pieces together and they don't always fit. And so, you know, it's, but when you, every now and then, when you have that insight, you know, you suddenly realize, oh, wait, you know, this result and that other result go together, and that means that X must be true. And then you test it and it is, you know, wow, it's really exciting. So I still, you know, after all these years, that's still kind of what drives me every day, is like I get up thinking, okay, you know, there's these things we're working on, and I'm trying to figure out how to, you know, how to answer those questions, or working with people in the lab on those challenges. And it's just, you know, it's fun.
Yes, definitely agree with the puzzle aspect of it, especially when you're talking about your joy in organic chemistry as puzzles, and it definitely follows through into our research careers, I think, as well. So our next question is a bit more fun. And so somebody wants to know if you think the movie Jurassic Park would be any different had they had CRISPR.
Well, I've had people ask me, you know, can the movie Jurassic Park be real? You know, can we actually do that now with CRISPR? And, you know, there are people like George Church that might think so, but yeah, I mean, I think, you know, CRISPR is a real boon for science fiction writers, isn't it? It's, you know, it's a fun technology to start imagining, you know, what can you do with this if you can really rewrite the code in any cell, if you can introduce new genes into organisms that didn't have them before, you know, can you create unicorns, and right, you know, all these kinds of things that you can imagine doing. And for the most part, those are science fiction, but they're certainly fun to think about.
Someone wants to know a little bit about your collaboration dynamics in your group. They want to ask, what do you think is most critical when managing a research group? And does your approach change based on the scale of the project or the collaboration?
Well, I guess over the years, I used to run my lab, in a way where, each person in the lab had their own project and was kind of working, relatively independently on their project, and talking to other people in the lab, but not really working in close collaboration with people in the lab or with anyone else. But about maybe 10 to maybe 15 years ago, I really started to change my style, and it wasn't really a conscious effort. It was more just changing because science was changing. The kind of science that we do in biological work on macromolecules, and where we need to understand molecular structures, but we also need to understand molecular dynamics, and we need to understand molecular interactions in a cellular environment, and all of those sorts of different kinds of questions that we wanted to be able to address or answer about the systems we were studying required a lot of diverse expertise. And so it became more the norm in my lab over those years for people to work in collaborative groups. And that's very much how my group runs now, is that I have groups of people in the lab that are working on projects together, and individuals still have a key aspect of that project that they're driving forward, so they have ownership over that part of the project, but they very much work in the context of a team. And I think it's made the science go better, I would say, and I think it's made it a lot more fun, really. I think it's, I think a lot of us, at least in science, enjoy the interactions of the lab. And it's obviously hard right now with COVID, and, people in my lab have commented on that. It's challenging when you don't have the same kind of face-to-face daily interactions that we were all so used to before. But even with the challenges, people have found ways to continue to work together and coordinate what they're doing, and I think that is really a hallmark of the kind of work that we do, honestly. And then I guess in terms of how I try to teach people in the lab, I guess I probably mostly try to teach by example. I really tell everybody in my lab that first and foremost, we are about great scholarship, right? About doing really the very best science that we can do, and really trying to understand, answers, the truth, whatever the truth is, and based on our data, trying to really figure out how things work. And as much as possible, setting aside our preconceived notions, and that's always a challenge, because we all get, have ideas that we become attached to, and we kind of like to see them be true, but sometimes they're just not, right? And so, we have to be open-minded as much as we can in our work. So I try to really, teach that sort of concept across the lab, as well as this idea of collaborative work styles. And we do a lot of work collaboratively with other laboratories. And in fact, we're now involved in, a few different larger consortia with multiple labs that are engaging on projects together. And, that comes with challenges, but in the end, I find that it's just really fun and really beneficial. And a lot of times ideas come out of those collaborations that I don't think would have come up otherwise. So it's, it's worth working through the challenges.
Definitely. So somebody would like to know, how has your life changed since this announcement of your Nobel Prize? Has anything gone better or worse than you expected, or have you gotten anything more than the guaranteed parking on campus?
Well, I've certainly been just really bowled over and blown away by everyone's extraordinary kindness. And, I've had, so many opportunities to connect with friends and colleagues, or, from everywhere, from my past, from my present. That's been just extraordinary. I don't have to tell anyone here, we're in an unusual year right now. In multiple ways, and it's been a hard year, and I do think that for many of us, having a chance to celebrate together and celebrate science, celebrate fundamental knowledge, celebrate collaboration, celebrate international collaboration, these are all things that I think for many people really resonate. So that's been really awesome. I don't know if it's surprising, but it's just been great to be able to share that emotion with so many folks. And then from talking with other colleagues at Berkeley who have, traveled this path before me, I understand that, in a typical year, there's a lot of additional travel that comes with an award like this, and, going to the White House, for example, things like that are just, for obvious reasons not happening right now. And for me, that's, there's two sides to that coin. On the one hand, it would be fun to be doing some of that. On the other hand, I'm kind of really enjoying, just being local and hanging out with my lab, and, being able to kind of carry on with our science in a way that, it might have been hard to do in a typical year. And I'm actually, really, I think one of the things that COVID has taught me is that you can get a lot done by Zoom. and maybe the crazy travel that I was doing kind of previously is maybe not a lifestyle that I want to go back to.
Thanks. The next person would like to ask how you have personally dealt with the lack of diversity in science, thinking primarily about the lack of women and people of color, and then what you think more broadly the scientific community needs to do to make much needed changes.
Yeah, great questions. First of all, I would just say that I've always approached science, I approached it as, I come at it first and foremost as I think of myself as a scientist. And I happen to be a female scientist, but I don't think of myself as a female scientist, if you know what I mean. So, and I think that's a principle that I think about a lot in the context of my lab and the collaborations that I have, because I think that the more that we can make science open to everyone and welcoming to everyone, and even more importantly, enabling to everyone. And what I mean by that is really empowering all the members of our scientific community to feel empowered to contribute to it. And I've seen this, in myself, honestly, right? Early on, feeling a lack of self-confidence, feeling like, who is little old me to speak up at a group meeting? I don't, what do I know? I don't seem to know, there's all these people that are much more experienced than I am, or more educated than I am, more knowledgeable about this topic than I am. But what I learned over time is that, many times, I can remember early on, when I was a student, I'd go to seminars and I'd be thinking about a question that came up to me, that I thought about during the seminar. And I would think, oh, I'm not going to ask that question because I'm sure it's a stupid question. And then lo and behold, somebody would ask it, right? And so then I would, and it would be, somebody famous, or somebody, somebody very, very prominent. And I would think, oh, maybe that wasn't such a stupid question. And I remember going to my first Cold Spring Harbor meeting back in the late 80s. And I think I was a second year grad student. And somebody was giving a talk about origins of replication in yeast. And this was a topic that I had actually written my qualifying exam proposal on. was my off topic and or my off not my thesis topic. And lo and behold, you know, one of the first questions at that Cold Spring Harbor talk was the key question that I was addressing in my thesis, my thesis topic. And so I thought, oh, okay, so maybe I'm not so crazy to think that I can do science, because I seem to be able to come up with questions that are worth asking and that maybe haven't been answered yet, and think about how to do them. And so I feel like I slowly gained confidence over time. And so one of the things that I try to do now in the lab is to talk about that and really welcome students, especially students that might be coming from a background like mine, right? And not a fancy, I didn't come from fancy schools at all. And really encouraging them to just trust themselves and trust their ideas, speak up. There's no question. There's no such thing as a stupid question. When you have a question, you can almost be guaranteed that other people in the room are thinking about it too, or if there's something you don't understand, it's probably because it wasn't made clear. And so I really try to create that environment, because I do think that that's a door to opening up STEM to a broader community, is for everyone to feel welcome and to feel empowered and enabled. And honestly, I think it'll make our science better, because we will have more people, more diverse people, and more diverse minds who are contributing to it.
Yeah, definitely. I think the more people that we have with diverse backgrounds will make everybody's lives better. And we need to welcome all those different backgrounds. As a graduate student, we're often given lots of advice. And sometimes we choose to or not to follow that advice. Is there any advice that you didn't follow and it turned out for the best? Has that ever happened to you?
Say that again, where advice did not turn out for the best?
That you didn't follow and that happened to be the best decision.
Well, and again, I feel like I tell some of the same stories over and over, but and I know I've told this one before, but I had a high school guidance counselor who once told me that girls don't do science, and I didn't follow that advice, and I'm really glad that I didn't.
Cheers to that.
Someone would like to know, when in your process of studying CRISPR, did you realize that it was going to be a really big deal?
Well, I think it was kind of it kind of came in stages, because I certainly had a feeling early on when we started working on it that it was certainly a very intriguing system, and there was nothing, really known about it when we got into the field. It wasn't really a field even, right? There were really just a handful of labs around the world working on it. So it was kind of this gut feeling that, this is something really interesting to try to understand. And then, so there was that. And then, when we were working on it in the early days, again, for me, I struggled a bit with, is this project going to remain a kind of a little niche project, a little, kind of a pet project in my lab with a couple of people, or is it going to get bigger? And, the first person that was working on it in the lab, Blake Wiedenheft, was a postdoc who was, a wonderful scientist, and also just a really fun, really nice guy. And so when, new students came to the lab, and I would say, well, what do you think you'd like to work on? They would often say, well, I'd like to work on whatever he's doing, because he was fun and he was a fun guy, and so that started to kind of build up a little group in the lab. And then eventually, when we started the collaboration with Emmanuel, I kind of did have the feeling that we were onto something really interesting with this double-stranded DNA cutting protein. And then it was when Martin Yinek, who in our lab, he was a postdoc who was doing the key work in that collaboration. I think when he figured out how to control the cutting activity of Cas9, how to program it with RNA, and importantly, how to simplify it compared to what nature had done. So taking a dual RNA guided protein and turning it into a single RNA guided protein that made it a two-component system that could be easily introduced into other cell types. That for us was really the key moment, I think, when we really looked at each other and said, wow, this is really, really exciting. And this protein could be an incredible tool for genome editing based on all the other work that had been done in that field. So it was kind of building up to it, and then this kind of light bulb moment where you say, wow, this is really going to be interesting and really exciting.
As you noted earlier in the talk, CRISPR can be used in a lot of scenarios, including human health, agriculture, or industries like synthetic biology. This question would like to know whether you think there is a second Nobel Prize winning research yet to be discovered that involves CRISPR.
Yeah, I mean, it's entirely possible. I mean, I think, CRISPR is a really enabling tool. So it's a technology that I think opens the door to a lot of types of experiments and answering questions that without it would be very difficult or impossible to address. So I certainly think that it's a tool that will enable new discoveries for sure. I mean, it already is. And will those discoveries be Nobel Prize winning? I wouldn't doubt it. I think it's entirely possible. Do I think that CRISPR as a tool is going to win more prizes? It could. I mean, I think there's the field, the tool development aspect of it is still very much very active and is moving forward. I think the thing to appreciate there is that fundamentally all of these CRISPR tools that have been developed are built around the fundamental chemistry of the system that is what Emmanuel and I focused on, right? This RNA guided mechanism that allows proteins in these pathways to interact with nucleic acids in a targeted way. And there's just really fundamental chemistry there that I think is very, very interesting and enabling, and does kind of enable all of these kind of different tool developments that have happened downstream. And I think that we're going to see continued advances there. And I, for one, I can't predict, where that's headed, but I certainly think that it's a very diverse toolbox that will continue to expand.
Even though you said that you can't quite predict the future of CRISPR, do you have any personal hopes for what CRISPR might be able to do for humanity in the long term?
Well, I guess I'm personally very excited about a couple of things. One is, and we've sort of already touched on these. One of them is really becoming a standard of care for curing genetic disease. I mean, it seems extraordinary to think that we might have such a technology, but I think CRISPR really does offer a path forward with many diseases that in the past were truly intractable. And because it is a programmable technology, it really is something that you can imagine being used in a personalized way. There's lots of challenges with that, of course, but I think fundamentally that is likely to be one of the big longer term impacts of CRISPR is really for correcting disease-causing mutations at the source. And then the other big one is sort of climate and the environment. And CRISPR might not be the first technology one thinks of when we think about tackling the challenges of climate change. But I do think that a combination of using it in plants and in sort of agriculture, crop sort of agriculture, as well as in microbes, soil microbes, microbes that are important for carbon fixation, for example. I think this combination could be very powerful as well, and that's something that we're quite excited about at the Innovative Genomics Institute, and partnering with our colleagues at UC Davis and LBNL and other partner organizations, where we have experts that are working in these types of systems, where they can actually benefit from and use the CRISPR technology to, again, do things that would have been very difficult without it.
Great. I'm going to bring a question a little further up that is still kind of on topic. So there's so many things that can be done, like you said, in health or agriculture, personalized medicine, carbon fixing bacteria. But in order to get there, there's a lot of hurdles that need to be overcome. So what are the major hurdles that CRISPR still needs to get through in order for it to become commercially or industrially or medically relevant?
I mean, I think a big one is delivery. How do we get these gene editing molecules into the cells or tissues where they are needed? And that's true whether we're talking about correcting a disease in a patient, curing a disease in a person, or whether we're talking about trying to make drought-resistant rice. The challenge does come down to delivery, and so that's something that we work on a lot at the institute, and have a number of there's a number of groups now that are focused in this area around the country and around the world, really, because I do think that's going to be very, very important for realizing the potential of CRISPR. And then there's, I think there's the broader question of what genes do we rewrite. We need to know more about genomes in general to know which genes to be targeting. It's one thing if it's a very well-studied disease like sickle cell disease, that disease has been investigated for decades. But in many cases, we have diseases that we know are ultimately derived from our genes, but the genetics are much more complex. And diseases like Alzheimer's and other neurodegenerative diseases come to mind. And so, is there hope for those kinds of diseases using CRISPR? I would say yes, but again, we really have to drill down to the fundamentals. What are the genetics of those diseases, and are there going to be ways to tackle them genetically using a technology like CRISPR? And by the way, the thing about CRISPR is that with all of the developments that have been made in the field, it's now possible to use it not only for making permanent changes to the genome, but also for making what we call epigenetic changes. So changing the output of the genome without actually changing the DNA sequence itself, by changing the methylation state of the DNA, for example. So that's something that I think is also a future potential of the technology, is to increase and enhance those capabilities so they can actually be used therapeutically.
Thank you. We have kind of another fun question. Have you seen the CRISPR beer from Temescal Brewery? Did you have anything to do with that? And if not, have there been any other sort of like fun random things that have popped up because of your Nobel Prize?
No, I haven't actually had that beer. Sounds like I need to.
I think Max has tried it, right? Is that what you said, Max?
Yeah, I think it was.
Was it good?
It was very good with CRISPR edited yeast.
Fantastic. Wow. Okay, I'll have to check that out. But I've definitely had, so people have sent me candy bars called CRISPR. I have a little Lego sort of little characters that are based on CRISPR. There are lots of things on YouTube that maybe folks have seen that are sort of animations based on CRISPR. So we're sort of lucky that it has it's kind of taken on a life of its own in some ways, and I actually think that helps with the the whole communication about it, for people to have ways to associate CRISPR with some of the underlying chemistry, even if even if we just ignore some of the details, but just thinking about what its activity is. I think it's been kind of interesting to see how that's playing out.
And I think we're getting towards the end of the student session. So maybe let's end on a positive note. I mean, we've always been on a positive note, but so in academia or science, we often, we fail, and we fail a lot. And this has come up over and over again in your anecdotes, is we always have to overcome failure. So how do you recover from your failures?
Well, I often, I try to take the long view, for one thing. I try to remind myself that, this is one challenge and out of many, and it's not the end of the road. So it's just, you move on at some level, right? You figure out if there's something you can salvage from the experience, and then you move on. And if it's something that, a failed experiment, or a paper that got rejected, or things like that, what do? Well, you just try to figure out if you can learn something from the experience. Sometimes you can learn something from it, and you try to take that away from it. And other times it's like, nope, just better to just move on, and and you just try to pick yourself up and keep going. I love a story. I have a great story about it. So when I was a postdoc, actually, in a lab in Colorado, there was a guy there who was making, one of my colleagues there, who was making a compound, and he was a chemist, and he was a real chemist, and he was in the hood every day making this compound, and it took several months, and as these things sometimes do, and it was a many-step synthesis, and he got to the very end, and he was on the very last step, and he had this thing in a round bottom flask, and he was lyophilizing it, and the flask had a star crack in it. Didn't realize that, and it shattered, and his final product went poof. And yeah, you can imagine, right? Some swear words were coming out of his bay, and he was not a happy camper. And so I tried to say a few comforting things, but he wasn't having any of it, and he left the lab. He was very upset. Understandable, right? Many of us have been there. I have had that type of experience. And the next morning I came to lab, and he was already there, and he had his goggles on and his lab coat on. He was standing at the hood, and I said, "Hey, what are you doing?" And he goes, "Well, I'm starting over." And I've never forgotten that story, because he just, he picked himself up, and he just got right back to it. And he made the compound and did the experiment, and wrote the paper, and it had a happy ending. But it really required some real mental toughness. And so sometimes I try to remind myself of that story, because it helps me get through those times when I too have to be mentally tough.
Thanks so much, Jennifer. We really appreciate your answers to all of these questions from students here. And I think especially for us grad students, it's really inspiring to hear from you and be able to listen to your experiences as a researcher. I want to pass this along to Matt, who's going to continue with the audience question portion. Thank you.
Thank you.
Thanks, Max. Thanks, Lydia.
Yeah, let me also thank both of you for emceeing that session. That was terrific. Thanks for asking the questions. So what we're going to do now is more of a live Q&A. So you should be able to enter questions in the YouTube chat, and those will be forwarded on to me, and then I'll ask those on your behalf. So just to start one out, one of the real leadership things that I think IGI did, and I know you were very involved in, was a lot of our COVID-19 testing on campus that we've been doing for the last few months. And I wonder if you could just speak a little bit about what was that experience like? There must have been a lot of politics, a lot of complications, and a lot of learning involved. So how did that all go behind the scenes?
Right. Well, it started back in March. So, like many of us, I was seeing this coming pandemic, and I had no idea it was going to turn out the way it did, by the way. But, you know, it seemed at the time like, okay, maybe this is going to be a couple of weeks that we're, yeah. But anyway, so, you know, back in early March, several of us at the Innovative Genomics Institute in our building, we got together and said, you know, what could we do to address this health emergency? And I was quite amazed that we put together, we thought, well, let's pull some colleagues together. We put out a little notice and just like, you know, I didn't advertise it that widely. We ended up doing it mostly by Zoom, and we had something like 60 people that came. And I realized that, wow, there's a lot of interest, and people are, you know, really trying to ask, you know, how can I get involved? What can I do to help? And one of the ideas that came up in that discussion was that you know, one of the most important ways that we can contribute right now to this emergency was with testing, because it was already clear that testing was desperately needed and there was going to be a shortage of you know, capacity. And so we started asking, you know, a few of us started asking, okay, what would it take to run a clinical testing lab at Berkeley? You know, could we actually do this? And as you alluded to, Matt, yeah, it was a real hairball of things that had to be figured out, and they had to be figured out very, very fast. And in fact, it went all the way up to Janet Napolitano, who was still the president at the time of the University of California. So we had to get approval from her office and her legal team to do it at Berkeley and get legal indemnification to be able to report results from patient samples. We had to get a CLIA license from our student health center. We had to get them to agree to work with us on that. We had to work with Dr. Ralph Green, who runs the entire university health system at University of California. He's based up at UC Davis. So we had to get him to get on board. And this all had to be done in a matter of days. And so I have to hand it to a number of our very senior colleagues. Bob Tejan really stepped up, and several other folks who just, you know, threw down the gauntlet. They spent, you know, tons of time on the phone with people, just, you know, working through the issues and trying to sort everything out. And bottom line was, 3 weeks later, we had a clinical testing lab that was up and running at Berkeley, which is kind of extraordinary. And since then, we've continued to operate that lab. That lab now has its own independent CLIA license from the state of California. We have our own clinical medical director of the lab, who is a volunteer who came in. He just was so excited about the work that the IGI was doing that he volunteered to work with us. Just extraordinary. It's a clinical pathologist who we don't pay, who just, you know, works volunteer. And then we have many partners in the community that we've worked with. We just recently signed an MOU with the university, our university, to provide most of the testing that Berkeley is now doing is being run at the IGI. And because we're running this lab in the context of our wonderful, you know, research university, we have lots of very smart scientists who are working in that lab and have been able to continually make the test better and better. We had the opportunity to develop saliva as a testing medium, and we have plans in place to start running a CRISPR based diagnostic test in the next few weeks that's coming online. So, you know, it's really been great to have that lab. And my hope for the future is that not only do we continue to provide testing that will help keep the campus safe and our community partners with their clients safe, but also to provide a way that all of our colleagues at Berkeley can work with clinical samples and do testing of other types of applications in the future. And that's something we're actively thinking about, by the way. So if there are people here listening to this that have ideas for what you might want to do in a lab like that, please reach out. I'd love to hear them.
Well, on behalf of all of us, thank you so much for your leadership in doing that, because it's been a really hard year for everyone on this call, but that's been a real bright spot, is the success of that program. And I think the campus owes you a lot of gratitude for that. Another question that we have from the people who submitted them in advance is, what's the funniest thing that happened to you in your scientific career?
Oh, the funniest thing. Well, oh boy. Funniest thing. I'm thinking hard here. The story that's coming to mind is something that happened not exactly to me, but I was in the lab at the time, and that was when a very good friend of mine, we were graduate students together, and in those days, you know, we had to make our own DNA and RNA oligos in the lab. There weren't companies that did that. We had to do it ourselves, and it was, you know, some real chemistry. We had to, you know, fill the reagent bottles. Not real chemistry, but anyway, we had these synthesis machines that we would run, and that was our lab job. And there was a lot of organic waste that would accumulate, you know, from these machines, and bottles had to be emptied. And one morning, I think it was like a Saturday in the lab, and I was in the lab with my friend, my colleague and friend, you know, running this machine. And then we had a new rotation student in the lab. And so my friend and I, both women, and the rotation student was a man. And so my friend picked up the bottle of waste, and the bottom just fell off, you know, and so this, like, you know, TCA, it was really gross. It was like TCA and just, I don't know what else was in there, but, you know, it was just like organic solvents soaked her clothing. And without even thinking about it, she ran to the shower in the lab, and she just took off all her clothes and pulled the shower. This is exactly the right thing to do, right? It's exactly the right thing to do. And and this rotation student, this man, you know, in the lab, he ran to his locker. You know, he had a locker down the hall. He ran to his locker, and he pulled out his sweatpants and a sweatshirt, and he came into the lab like this. He was walking down the hall like this, holding out his sweatpants for her to put on. I thought it was so sweet. It's funny in retrospect, not so funny at the time, but he did join the lab.
Thanks for sharing that story. One of the other questions we have is more from the undergraduate perspective about what advice you would give undergraduate students who are seeking opportunities to join or work with research groups.
Be persistent. That's my advice. And the other piece of advice that's effective in a lab like mine is get to know the members of the lab. A lot of times, you know, the faculty are running around, you know, chairing departments or, you know, teaching classes, all the things that we do, and we're not so fast on our email. So I really encourage undergraduates to get to know members of the lab, and a lot of times a good way to do that is through your GSIs, and find out, you know, what labs they're working in and what they're working on. Ask questions. And then, you know, ask them if there are opportunities to do research in their lab. And in many cases, the students that I've had, undergraduates that have come to my lab, have come through that path, you know, and they end up working with somebody that they met through a course they were taking, and they hit it off, and then they end up working together on a project in the lab. And that can often work very well.
Thank you. So one thing everyone knows about your scientific success, but one thing people may not know as much about is that you're a very dedicated parent. And so I was wondering if you could talk about how you balance, or what tips do you have to balance the work-family life?
It's always a challenge. I always feel like I'm juggling. Not so much now. I mean, my son is now 18, and, you know, he's a freshman at Berkeley, and he knows more than I do about most things. So, yeah, I mean, I think, you know, all along in our family life, and one of the things that we've done, and, you know, I'm sort of lucky that, you know, I'm married to another very distinguished Berkeley faculty member, Jamie Cade, who's a member of the chemistry department as well. And so we've often, over the course of our time as parents, we have found ourselves, you know, traveling to scientific conferences with our son. And, you know, we just did that from very early on, even when he was a baby. You know, we would take him to conferences, and, you know, it meant you had to kind of, you know, you weren't 100% at the conference because you had to, you know, be spending time with your child. But boy, was that worth it, because I think our son really, he grew up kind of knowing that science was really important to both of his parents. and that it was something we both felt really passionate about and really dedicated to. I can remember, teaching a class. My son was a baby. He was just a few months old, and I had him in a stroller, and I literally strolled him over to class and had somebody sitting outside watching him while I taught the class. And then I came out of class and took him back to my office in the stroller, that kind of thing, right? So you just have to be creative, and you find ways to work it out. And I'm a firm believer that it's actually good for kids to see their parents in their, see their parents' passions, their professional interests. I think that's important, because I think my son certainly has grown up knowing that, his mom is, I hope he knows, I think he knows that, his mom is really, he's number one, I'm really dedicated to him, but that I'm really passionate about my work and dedicated to my students and what I'm doing. And that's an important part of my life and who I am. And I want him to know that about me. And I want him to know that about, future partners that he might have, right? And knowing that, it's important to value that.
Thank you. So as a parent for a youngster myself, it's an inspiring example. So one thing that I hear people say a lot, especially graduate students as they're moving through sort of their second, third year, is they start to think about the future, and they start to think about whether they want to be an independent scientist doing research at university, perhaps. And one of the more common things I hear people say is they just don't think that they have good ideas, that they don't think that their ideas are going to be exciting or worthwhile to pursue. So what was your own experience like with that? And kind of when did you reach that transition where you realized your ideas had merit?
Well, I can remember feeling that same way. I mean, I remember, when I was in graduate school, looking at my advisor and, his, writing grants and, realizing, wow, he's responsible for a lab of, it was like 25 people, and thinking to myself, oh, I could never do that. Just, wow, I couldn't imagine doing it, honestly. And also, yeah, this fear of, would I ever have a good idea? Would I ever have an idea worth pursuing? But the thing was that, I just, for me, again, it was always, I always just focused on what I thought was really interesting. And when I was finishing up graduate school at that time, there was really no work that had been done on the structures of RNA molecules. And I could see that, that was kind of the area I had been working on, but doing more, sort of biochemical experiments. And I could see that to make progress in the field, that was going to be essential, was really to understand, first of all, did RNA molecules generally have defined 3D structures? Nobody really knew at the time. And secondly, if they did, what were those structures, and how could that help, how could we understand what those architectures might tell us about the chemistry of, catalytically active RNAs, which again, that was where I was, focusing when I was a graduate student. So it was really kind of just driven by that curiosity. And I still remember, when I decided to go into that as a postdoc, I remember thinking to myself, well, this is it for me. I'm probably going to never be heard from ever again. Because, there was a good chance that wouldn't work out. I mean, nobody had really done that before. And, there were a lot of people saying, well, this really won't work for all sorts of reasons. It sounded really good, but I was very stubborn. And I thought, if I don't try this, I'll never know. And if I try it and I fail, then at least I'll know that I gave it a shot. And so I just felt like I have to try it. And, believe me, there were many moments when I thought I was going to fail, right? I thought it wasn't going to pan out. And I was 2 years into my postdoc, and I saw all these people around me in my lab, publishing beautiful papers, and it wasn't me doing it. And I thought, yep, that I was right. No one's going to ever hear from me ever again in science. But I kept going somehow. And, and then, little things would happen that kind of kept me going. I got a crystal. Right? We got a crystal of RNA, and I thought, wow, so RNA can crystallize. This is really interesting. And, and then we had to solve the phase problem. And then there was the chemistry behind that. And it was a puzzle, and I was stubborn. I'm like, I'm not going to let this thing defeat me. I have to figure this out. So I don't know. I just think that for, getting back to your question, I think I really encourage students just to really figure out what it is they're passionate about and pursue that. Because a lot of times if you have a question about something in your mind, or something that you're wondering about, that's gonna drive your desire to figure it out. And if you really, are stubborn about it and you're just dedicated to it, you're gonna get to an answer. And you have to be open to, where that science will take you.
Yeah, thank you. And then as one final question asked by a member of the audience, so thinking ahead, so clearly there's a lot of things to be discovered with the CRISPR-Cas9 system, but where do you look for new inspiration for new research projects and directions?
Well, these days, a lot of where I look is to nature and especially to microbes, because I mean that's honestly where CRISPR came from in the beginning. And I've come to appreciate, especially through our interaction with Jill Banfield and many of our other colleagues working in that space, that wow, there's still a lot that we just don't understand about these organisms. Most of the organisms themselves aren't known, and when you sequence their genomes, as Jill Banfield's lab does, you find lots and lots of genes and systems of genes that are just completely uncharacterized and unidentified. So I think there's a lot of fundamentals to be done there. So that's for me, I think still an enduring, fascinating mystery is, kind of what's out there and how do we investigate it?
It's such a good example of the important role of basic science and how you just have to pursue it. And sometimes amazing things happen that change the world. So well, that brings us to the end of our session today. But again, let me thank you, Jennifer, for spending the time with us and answering all of our questions and continuing to be an inspiration to all of us. I also want to thank Lydia and Max once again for leading all the questions and the discussions. And then also there's a lot of behind the scenes people who made all of this work, all the technology and the questions and feeding those. And I want to thank the whole development office who put together this great event. Finally, thanks everyone for joining us this afternoon. Be well, be safe, and we'll talk to you later. Bye.
Thank you all.